Hello everyone, welcome to my presentation. I'm Aparna Annamraju. I'm a first year PhD student with FWF department. So the title of my presentation is Switchgrass Ash Content Relationship with Cellulose, Hemicellulose and Lignin. So what is switchgrass? Switchgrass is a grass plant that is grown during warm season. It is a leading bioenergy crop, easily grown. In US, it is actually grown in Midwest, South and Southeast regions. So I mentioned the term bioenergy crop, right? So what exactly is a bioenergy crop? You can classify a crop as a bioenergy crop if it can be grown with low maintenance and low cost, and it is efficiently used in the production of bioenergy. Two main types of crops come under this category, woody and herbaceous. Switchgrass comes under the category of herbaceous crop. Other examples of dedicated bioenergy crops are hybrid poplar and willow. So what exactly is bioenergy and why do we need it? So this is a graph I obtained from EIA website. On the x-axis, it shows the usage of fuels over the years, and on the y-axis, the amount we have been using. The three main fuel sources for us are coal, natural gas, and petroleum. But down here is biomass, and that is the part that I'm interested in looking at. And why is that? That's because in the year 2015, the data is shown that the renewable share of energy was largest at almost 10%, whereas the fossil fuel consumption made up only 81.5%, which was the lowest in the past century. So it is worthwhile to invest and learn about this biomass and bioenergy crops. But besides the utilization, besides the increase in demand for the bioenergy, the other aspect is, if you look at this picture which I obtained from the billion ton project, the three key aspects that will help due to this bioenergy revolution is: one is the reduction in the greenhouse gases, two is the decrease in the imports of foreign oil, and the third factor is rural economic development. These three aspects are highly impacted by understanding the bioenergy concept. So where exactly these components of interest for us are present in these bioenergy crops? The main part where these components are present is called as plant cell wall, and in this plant cell wall, the three main components, and each of which can be utilized in some form of bioenergy, are cellulose, hemicellulose and lignin. And this picture actually shows the structure of the three main components present in the plant cell wall. As you can see, cellulose is a polymer composed of glucose units. Hemicellulose is also a polymer composed of 5 sugar and 6 sugar units, whereas lignin is distinctly different from the other two components. It is mainly made up of aromatic benzene rings. So it's been shown that all these three components can be used in the production of bioenergy. So the lignocellulose components are present in switchgrass too, but what other things are present in when we are looking at the biomass of switchgrass? So the glucan, xylin and galactan, they're all part of hemicellulose. Even arabinin and mannan, Lignin, like I showed you in the previous slide, is mainly made up of aromatic rings. But two other things that are shown in this graph, x-axis, are extractives and ash content. Ash content is obtained from biomass once you incinerate it at 575 degrees, very high temperature. It represents the mass of material that cannot be converted into any utilizable part of bioenergy. And extractives are composed of basically the chemicals that are used um, during the process of growing of these bioenergy crops. We need to have a good estimate of the amount of extractives present in the biomass and the ash content of the biomass in order to know what is the true amount of biomass that can be utilized for bioenergy aspect. One more point I forgot to mention on this slide is extractives can be easily removed by doing a pressurized hot water and ethanol extraction, and you can get a good estimate of that amount by that process. And ash content, like I mentioned, it's by incinerating your biomass at really high temperatures. Usually it's around 575 degrees Celsius. So this is how my data looks. So when Dr. Zobel mentioned that we are supposed to present a paper during this course, the kind of work that I do is mostly at looking at the utilization of the components of biomass into a bioenergy. That would be cellulose, hemicellulose, and lignin. So since I started recently, just in spring, one of my lab mates was kind enough to give some data to me. So what are we looking at is different switchgrass types that were grown in one particular field, and the ash content, extractive cellulose, xylem is also part of hemicellulose, and the total carb lignin content of each of these switchgrass samples is shown, and I have 84 such samples. So the first thing you can see from this data is it is distinctly different from what Dr. Zobel discussed in the class. I do not have the kind of typical forestry or wildlife data. So from the data analysis point of view, the one thing that I could do was do a series of linear models of ash content with cellulose, ash content with hemicellulose, ash content with lignin, ash content with xylem, trying to understand how ash content is affected, how ash content actually affects the amount of these components present. Here I'm showing you the sample of relationship between ash content and cellulose. And like I said, I discussed with Dr. Zobel based on the data, because I have a rather simple data where it shows each row in my data are the numbers associated with that particular switchgrass. In my paper 2, all I did was a series of linear models and ANOVA analysis, trying to understand the relationship between ash content and each of these biomass components. And here is the ANOVA result for cellulose. And further, to understand how they are related, I take correlation, simple Pearson correlation, and it shows that the ash content and cellulose have a strong negative Pearson correlation. And I did similar linear models for hemicellulose, lignin, total carb, and xylem, which I did not show in this PowerPoint presentation. Here is a summary of data from paper 2. I'm showing only the Pearson correlation values, which help us understand how they are related. So the more ash we have, generally what I have seen from the data is there's less cellulose present. And from bioenergy point of view, we need to have more cellulose so that we can produce ethanol from it. And using my ANOVA, actually, I could establish that the there was 85% predictability between these two variables us of association, trying to explain the relationship between them. For my alternate approach, I try to think hard of how to address my kind of a data, which is really different from typical forestry data that Dr. Zubel showed in the class. And all I could think of was to do a non-parametric test. So this I tried to do in R, but somehow I kept getting an error. So I followed his suggestion and I did it in SPSS, where I tried to create two groups of data, one with ash content 4.2% and above, and the other one had lower one. So trying to understand, again, this is simple correlation, trying to understand the relationship between how ash content and cellulose are related. The conclusions that I can make about my data and the kind of approaches that I took are, I feel that the linear models do a decent enough job to explain the relationship between ash content and each of my cell wall components. For alternative, like I mentioned, I tried a non-parametric approach. But given the sample size, I have 84 different kinds of switchgrass samples from the field. I feel that the parametric tests did a better job of explaining the relationship between the ash content and the biomass components. My acknowledgements, my lab mate Chu Hamilton, who gave me the data, and my supervisor, and here are the references. Thank you so much.